In chapter 30, we'll talk about male genital and reproductive functions. So we're going to start with this picture here, and what we're looking at is basically a mid-sagittal cut through the pelvic area. And what we can see is basically, the scrotum around here with the male gonad inside, which is the testy. You can see the erectile tissues of the penis, as well as the urethra that travels through it. We can see some other internal pelvic organs like the urinary bladder, our rectum, the prostate, and seminal vesicles back here. But let's go into more detail on the scrotum right here. So on the scrotum, the outside of this is basically just skin. It's thin skin, hairy skin. And deep to that, what we're going to find then is in the wall of the scrotum, you're going to have some different muscles like dartos and cremaster. And what these muscles do is they help regulate the distance of the testes to the abdominal cavity or pelvic cavity to help regulate the temperature inside the scrotum. It turns out that sperm, as they're generated within a testes, need to mature at a lower temperature than internal body temperature. And so what helps regulate that temperature then are the muscles you find within the wall of the scrotum here. Now, each testes has an attached epididymis, and the epididymis has a head, body, and tail portion. And basically the epididymis is just a storage site for sperm. So once sperm have matured, they can get stored in the epididymis until they're used or released. And the epididymis is also what attaches to the vas deferens or ductus deferens. So vas deferens is a muscular tube that goes up through something called spermatocord. It goes up into the inguinal ring here, through the inguinal canal, into the pelvic cavity, where it wraps back around the urinary bladder, and then the vas deferens meets up with a gland here called the seminal gland. So the seminal gland is found in the posterior part of the urinary bladder, and it produces the majority of the volume of semen. But that sperm, or we call testicular fluid, mixes with seminal fluid, and those enter the prostate gland through something called the ejaculatory duct, which is right here. Now, the prostate gland is also a type of gland that is involved with creating various secretions that are incorporated into semen as well. And that also converges with the urethra from the urinary bladder. So beyond the prostate, what we find that is the continuation of the urethra. There's some muscle here just inferior to the prostate, which we call the urogenital diaphragm. It's a piece of smooth muscle that can help, it's a sphincter that prevents, the release of urine or semen. Now, the name of the urethra right here by the urogenital diaphragm, we call the membranous urethra. And the gland that's nearby here is an additional gland called Calpers Gland or bulbal urethral gland. What Calpers Gland does is it creates a clear kind of viscous secretion during sexual arousal that helps neutralize the acidity of the urethra. Earlier we talked about how it's acidic urine that helps kill off bacteria. Well, that potentially acidic urethra could also damage sperm as they travel through urethra. So it's the bulbal urethral gland here that helps neutralize that urethra. So then semen's going to leave out through the penile urethra, which continues on through the erectile tissues of the penis until we get to what we call the external urethral orifice, which is the very tip here. Now, there's two main erectile tissues to the penis. We got corpus cavernosum, which is the largest, and it sits more sort of anterior or dorsal on the penis. And then we have corpus spongiosum, which is a little bit more posterior or ventral on the penis right here. And that's why it's called the spongy or penile urethra, because the urethra goes through corpus spongiosum. And the purpose of corpus spongiosum is to basically help maintain the patency or openness of the urethra during an erection. That way, semen can leave. Now, in this slide, what we're looking at is a cross-section of the scrotum. And so you can see the wall of the scrotum, like we mentioned earlier, has an outer layer of skin or integument. Deep to that, what we find is a muscle called dartos muscle. Dartos is a smooth muscle that you find in the subcutaneous tissue of the scrotum. And dartos and cremaster together help regulate the distance of the testes to the pelvic cavity in order to regulate body temperature. So, for instance, if the internal temperature inside the scrotum is too warm, dartos and cremaster will relax, and they basically help the testes descend to cool off. And if it's cold, dartos and cremaster will contract and help ascend the testes up towards the pelvic cavity in order to warm up the inside of the scrotum. That way, sperm can mature easier at that ideal temperature. Now, what we find is that each testes, which we're just kind of looking at one here and one there, each testes is supported by a type of tissue which functions a lot like serous membrane. So remember, serous membrane was a lubrication type of tissue that reduced friction. Well, even the testes are surrounded by something that functions like serous membrane. It's called tunica albuginea. And what we find here is that it actually surrounds the testes, and the testes then are supported by a nice fluid layer, which we call the scrotal cavity. And within that, we'll have scrotal fluid, which is a lubrication type of fluid that prevents friction around each gonad or testes. Now, within the testes, what we find that are different lobules. So each of these little lobules are separated by septa, and these septa are basically infoldings of tunica albuginea. And within each lobule, what we find that is these big old seminiferous tubules, which are basically tubes of cells that have a duct where sperm then, after they've divided, can actually travel towards this little area here, which we call the rete testis. And then the rete testis will converge with the epididymis. And the epididymis is basically the storage site for sperm. So if we look at a cross-section of a seminiferous tubule, that's what we're going to see here. And what we'll find then is that the seminiferous tubule is made of lots of different cells that are in multiple layers here. And we have things like nurse cells that basically help nourish and support the dividing spermatozoa. Spermatozoa here are the mature sperm cells that are basically, they start to look like sperm. And those spermatozoa come from spermatocytes, which you can see here. And those spermatocytes will divide through a process called meiosis. And meiosis yields a mature gamete, which has half of the number of chromosomes. So it has a full set of copies, but half of the number of copies. That way, when a sperm fuses with an egg, then you have a full set of duplicate chromosomes that can make a new organism. Now, outside of the seminiferous tubules, what we have are these interstitial cells, and these interstitial cells are the ones also called Leydig cells, but these are the ones that release testosterone to help support the growing spermatozoa. Now, the process of meiosis in the seminiferous tubules we call spermatogenesis. And so we find that as we start out with spermatogonium, which is diploid, which means it has a complete set of chromosomes that are also in duplicate. And these can divide through a process called mitosis and to make two copies. We keep a daughter cell that stays behind. And then we have a primary spermatocyte, which is still diploid, that can divide further through meiosis to make spermatozoa or sperm cells. So what happens then is this primary spermatocyte here can begin to divide in meiosis. And when it divides, what happens then is that these chromosomes get separated with 2 rounds of cell division. So from one cell, you get 4 cells, and each of these cells has half of the number of chromosomes, but it has a complete set of copies, just a half of the number of chromosomes. So we call that haploid because it has half of the complete set of chromosomes, but it has the correct number of copies. So we have 23 copies of chromosomes, but not 46. We have 23 chromosomes and 23 copies. So these can mature then through a process called spermiogenesis into what a sperm cell looks like. And basically what these sperm cells are comprised of is a head, a body, and a tail. The head group is really just what contains, the genetic material as well as a modified lysosome called an acrosome. In the midpiece or body, what we find that is a coiled mitochondria that can produce ATP that can aid in the movement of this long tail, which is made of a flagellum. And this flagellum can use the energy derived from the mitochondria to basically help propel the sperm in a particular direction. So looking at the structure of a mature sperm cell in more detail, we find our head, our midpiece, and our tail. Now the head has a nucleus, and the nucleus there would have your 23 chromosomes. And the acrosome is the very tip of the head group here that functions to basically help digest the outer surface of the egg, because this acrosome is full of digestive enzymes and acid to help break down the outer layer of the egg. Now, we find that in the midpiece, we have a spiraled mitochondria that can derive some of the chemical energy from sugar you find in semen to help convert that to ATP to power this long flagellum. And the purpose of this flagellum is basically cellular propulsion, or to move this sperm cell in a particular direction. Now, we know that sperm are stored in the epididymis, and that during an ejaculation, the sperm are basically moved from the epididymis up through vas deferens, and vas deferens then goes through the inguinal canal back behind the urinary bladder, where it meets up with the seminal vesicle here. Now, the seminal vesicle or seminal gland makes the majority of the volume of semen. It's about 60% or so, and basically it's a slightly alkaline and fructose and glucose rich viscous solution that gets mixed with our testicular fluid here to help form basically semen. And those converge at the ejaculatory duct into the prostate. Now, the prostate also secretes prostatic fluid, which is also going to be rich in things like sugar, fructose, but it has something else called prostate-specific antigen, or PSA. And PSA is an enzyme that helps liquefy semen. So it helps basically make semen more liquid and less viscous, which makes it more easy to kind of flow around. Now, the prostate also secretes prostaglandins, which we learned about were involved in the inflammatory response. So during inflammation, they helped increase blood flow. But if you'll remember, prostaglandins also stimulated smooth muscle contractions in blood vessels nearby to cause turbulence that can promote blood clots. Now, the prostaglandins that are incorporated into semen by the prostate gland help with vaginal contractions, because once semen enters the vagina, what happens is smooth muscle in the wall of the vagina can start to contract through peristalsis, and it helps propel that semen up towards the uterus. Beyond this, what we find then is this is the urethra, the beginning of it. We call this the prostatic urethra. We have our bulbal urethral glands, which make a very small volume of semen, but this secretion is mostly going to occur prior to ejaculation, and it's a kind of clear, viscous type of secretion that helps neutralize the acidity of the urethra. We find these near the urogenital diaphragm, which is a little layer of smooth muscle that functions like a sphincter to prevent unwanted or unnecessary passage of semen or urine. Now, for the erectile tissues of the penis, what we find here then is corpus cavernosum and corpus spongiosum. Corpus cavernosum contributes to the majority of the erection, because it's going to be a spongy tissue that engorges with blood during sexual excitement. And it turns out that sexual excitement is mediated by the parasympathetic nervous system. So during rest and digest, when you're parasympathetically active, then these erectile tissues are more likely to engorge with blood during sexual arousal. But some of this is also a reflex that doesn't involve conscious thought. So not all sexual arousal is due to conscious stimulation. Now, corpus spongiosum is actually a little bit more posterior, or what we call on the ventral surface of the penis. Now, this is this is also an erectile tissue that surrounds the urethra, and it continues up into the glands of the penis or the head, and that's also going to be the type of erectile tissue you find in the head of the penis here. Now, it's these erectile tissues together that will engorge with blood to form an erection. Now, looking at these erectile tissues in a different view, we can see corpus spongiosum that surrounds the penis, corpus cavernosum on the outside of that, and blood flow to the penis is increased during the parasympathetic response, where nitric oxide helps vasodilate blood vessels that serve these erectile tissues. These erectile tissues, like a sponge, start to engorge with blood. This increases pressure, which contributes to the erection. And we also have these venous outflow valves that prevent blood from leaving the erectile tissue too quickly. That way blood can build up and form pressure. Now, On the last slide here, I'm sorry, there's another view of the erectile tissues as well. So corpus cavernosum, corpus spongiosum, which surrounds the urethra. Now, on the last slide here, we're going to see is some of the hormonal influences of things like sperm growth, as well as other peripheral effects of testosterone. So we know that the hypothalamus releases gonadotropin-releasing hormone, and this GnRH, or gonadotropin-releasing hormone, can travel through the bloodstream to the anterior lobe of the pituitary gland, where it stimulates some endocrine cells in the pituitary gland to release FSH and LH. FSH is called follicle-stimulating hormone. LH is luteinizing hormone. And although these are named with respect to female reproductive function, they still play a role in male reproductive function as well. So FSH in males stimulates these nerve cells. Remember, nerve cells are the ones that help basically promote sperm growth. And they do this by making things like androgen binding proteins, as well as helping to form what we call the blood-testes barrier. Now, once these nerve cells are active enough, they release a hormone called inhibin, which is involved in negative feedback to inhibit things like GnRH and FSH. That way they're not too overly active and you don't produce sperm too quickly. Now, these are going to promote basically spermatogenesis and spermiogenesis. Now, on the other side here, we have luteinizing hormone, or LH. And what LH does is it stimulates these interstitial cells. Remember, those are the ones that aren't found in the seminiferous tubules, but are found outside of those tubules. And these interstitial cells release testosterone as a result. Now, testosterone is the androgen type of hormone here that plays several different roles. For one, testosterone can stimulate nerve cells as well to also promote spermatogenesis and spermiogenesis. Testosterone does have some peripheral effects. For one, it helps maintain male libido or sex drive. It stimulates bone and muscle growth. It also establishes male secondary sex characteristics, like deepness of voice, body hair in certain areas, other characteristics that you associate with being more male. It also helps maintain accessory gland organs of the male reproductive system. So maintains the activity of like your prostate, the seminal gland, and bulbar urethral gland. Now, testosterone also plays a role in negative feedback, so that once testosterone levels get high enough, it actually inhibits additional release of GnRH, which also would lead to the underproduction of FSH and LH. But this is important because you don't want too much testosterone, because then you have excessive peripheral effects and excesses in sperm growth. So that testosterone plays an important role in negative feedback by inhibiting the additional release of hormones that not only promote more testosterone release, but also sperm growth. So that's that negative feedback process here.